A very good afternoon to all. So I'm launching the technical poll. Quickly answer through poll. My audio is clear and my screen is visible. Okay, so today we are going to discuss principle of inheritance and variations. So before we proceed with the lecture, let's discuss some of the terminology regarding the principle of inheritance and variations. First thing is the genetics. Genetics is the study of genes and chromosome. Inheritance is the transmission of character from the parents to the progeny. Okay, and it is the basis of heredity. If we are talking to the recent generation, then we are referring as inheritance. But if we are talking about the several generation, we refer it to hereditary. So transmission of character from the parents to the offspring is known as inheritance, and the degree by which the progeny differs from the parent, that is known as variations. For example, we are different from our parents. We are not identical to our parents because of the variations. Now let's discuss the character, a heritable feature among the parents and the offspring. Example, the eye color. So the character is the eye color, and whether the eye color is brown, blue, then it is known as a trait. For example, we are going to study this in Mendel. The Mendel has taken 7 characters and 14 traits. So the variants of the characters are known as trait. Allele are the alternative forms of genes or the molecular form of genes, and the dominant allele, we represent the dominant allele with the capital letter and the recessive allele with the small letter. So if the character which is expressed in the heterozygous condition, then it is referred as a dominant character. And if the character which is suppressed in the heterozygous condition, it is known as a recessive characters. Okay, recessive character you can only see in case of when there's a homozygous recessive condition. When the recessive alleles are present in a homozygous condition, then only you will able to observe the recessive character. Phenotype is a physical expression of a character, okay, that you can easily observe with the eyes. That is the phenotype. Genotype is the genetic constitution of a character. For example, if this is a tree, this tree have a phenotype tall, and it could have a genotype this homozygous dominant genotype or a heterozygous dominant genotype. So this is the genetic constitution of this organism or the plant, and this is the phenotypic representation, that it is tall. You can see it with your eyes. Hybrid, when the individual is produced by the mating of a genetically unlike parents. Now let's discuss the experiment which is conducted by the Mendel. Mendel conducted the hybridization experiments. He performed the cross-pollination on the pea plant for 7 years. Remember this, that he worked for 7 years, and remember this year. It's very important. Remember the year. Okay, so what Mendel did, he took 7 characters and 14 traits. These traits, out of which the 7 are dominant and 7 are recessive. And these traits, they are opposite and contrasting. For example, here the first character is height. He has taken the first character is height, and the traits are tall or dwarf. Okay, these traits, they are opposite or the contrasting. For example, tall, opposite of tall is dwarf. Second character he has taken is the position of the flower. And the traits are axial or terminal, that the position of flower could be axial, could be present at the top, present at the axis, and the terminal that the flower could be present at the top, that is at the terminal position. Third character, he took the flower color. Violet and white, they are the trait. Violet is dominant, white is recessive. The fourth character he has taken is the shape of the pod, full or constricted. Fifth character he has taken is the color of the pod, green or yellow. Sixth character he has taken the shape of the seed, round or wrinkled. So round and wrinkled, they are the trait. Seventh character is the color of the seed, yellow and green. And so total he has taken like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So there are 14 traits and 7 characters. Okay, 7 characters are height, position of the flower, color of the flower, shape of the pod, color of the pod, shape of the seed, and color of the seed. So remember these all the traits and the characters. These are very important. Okay. And Mendel, he performed the cross hybridization or the artificial pollination on the pea plant, and especially on the true breeding lines. True breeding lines, for example, if this plant is tall and having a genotype homozygous dominant, that is capital T, capital T. If I cross it with the parent having the same genotype, then the F1 progeny, it will produce a gamete capital T. It will also produce a gamete capital T. So F1 progeny will have this, like all the offspring will be tall and they are homozygous dominant. If we carried out the selfing of the F1, that is capital T crossed with capital T, in the F2 generation, we are also getting the capital T, that is all tall. So true breeding line represent that it is undergoing the self-pollination and the trait becomes stable. For example, this tall, this is the trait, and it becomes stable in the generation. For several generations, this trait is stable. So this is known as the true breeding line. So we also call it as pure lines. And why the Mendel uses the pea plant? First is because the pea plant completes its life cycle in one season. It's an annual plant. Second, the pea plant, it produces a large number of seed. Third, the contrasting traits like tall, dwarf, axial, terminal, these are the contrasting traits. They are clearly visible in the pea plant. And pea plant is a bisexual flower, so it can show self-pollination as well as artificial pollination can be done in the pea plant. So that is why the Mendel chooses the pea plant. Moving next, Mendel performed 2 cross. One is the hybrid monohybrid cross, and the second one is dihybrid cross. So first we are going to discuss a monohybrid cross. Mono means one. Okay. So in the monohybrid cross, one character is studied. And according to the Mendel, one character is controlled by one gene. Okay, but Mendel named this, later we call it as gene, but Mendel call it as factor. So according to Mendel, one character is controlled by one factor or one gene. So that means we are going to study the inheritance of one gene. So the character here is plant height. Height is the character. And the trait are tall or dwarf. First, he took the homozygous plant. First, he took a pure line because they are homozygous. And we are representing these factors like we are representing capital T in pair, or capital T, small t in pair, small t, small t. Why we are writing it in pair? Because plants, they are diploid organism. In diploid organism, we write the genotype like this in pairs. So first, he took the plant. Here, the plant is homozygous tall or the homozygous dominant, that is capital T, capital T. Always remember that we represent the dominant allele with the capital letter and the recessive allele with the small letter. And another parent plant is a homozygous dwarf, that is small t, which is a homozygous recessive condition. So this plant produces the gamete, which is capital T, and this plant produces the gamete small t. In the F1 generation, we are getting capital T, small t, that is heterozygous tall. because the dominant allele will going to express and the recessive allele will not going to express. So in this condition, we are getting the tall plant because the dominant allele, which is capital T, is going to express and show the its a phenotype. When we do the selfing of the F1, we obtain that. So here the selfing is taking place. We have self the heterozygous tall with the heterozygous tall, and it produces two gamete. The 50% is gamete is carrying capital T and the 50% of the gamete is carrying small t. Here also 50% is carrying capital T and 50% carries small t. And when we carried out the cross, we obtained that there's homozygous dominant, there are two heterozygous dominant and one homozygous recessive or dwarf. So after crossing, we are obtaining the phenotypic ratio 3:1, where 3 are tall, 1, 2, 3. 3 are tall and 1 is dwarf. And the genotypic ratio, capital T is to capital T, small t is to small t, small t, that is 1:2:1. So this is the genotypic ratio and this is the phenotypic ratio. Please remember this ratio, that we are obtaining this ratio in case of monohybrid cross. And this graphical representation is known as a Punnett square. So Punnett square is the graphical representation where we calculate the probability of all the possible genotype of the offspring. One more important point, that on the basis of the monohybrid cross, Mendel, he proposed two laws. Remember this, that on the basis of the monohybrid cross, Mendel proposed two laws. One is the law of dominance and another one is the law of segregation. So he proposed two laws on the monohybrid cross. So law number one says law of dominance. So according to the law of dominance, if there's a dissimilar condition, for example, if there's a capital T and small t, we're getting a heterozygous condition. In that case, the dominant allele will express and the recessive allele will not express. Okay. So this is a condition of the phenomena is known as dominance and the law is known as a law of dominance. But this law is not universally applicable. Remember this, that the law of dominance is not universally applicable. Second law is the law of segregation. And this law of segregation states that during the gamete formation, for example, this is a parent. So during the gamete formation, one gamete will carry only one factor or one allele. For example, this gamete is carrying only capital T and this gamete is carrying only small t. So one gamete will carry only the one allele during the gamete formation. So this is known as the law of segregation, and we also call it as the law of purity of gamete. Remember that this law of segregation is universally applicable. Okay, this law is universally applicable. Moving next is the test cross and the back cross. For example, this is a plant. This plant has a phenotype tall. And this is a dominant phenotype. Okay. And this plant on crossing, it produces a progeny. That 3 are tall and 1 is dwarf. Okay. But we don't know the genotype of this plant. We don't know whether this plant is having a homozygous dominant genotype or it is having a heterozygous dominant genotype. So to calculate or to identify the genotype of the plant having a dominant phenotype, we call it as test cross. So what we do, we cross this plant with the parent having a homozygous recessive condition. Okay, if we cross the F1 hybrid with the homozygous recessive parent, this cross is known as a test cross. And why we are performing this test cross? To identify or to calculate the genotype of the dominant phenotype. Okay. For example, I know that this plant on crossing is producing 3 tall and 1 dwarf. So if I cross capital T and small t with small t, it will produce 2 gamete. It will produce 2 gamete, capital T and small t, and this will produce 1 gamete, small t. After crossing, so I'm getting tall and dwarf. That is 50% is tall and 50% is dwarf. That is 1:1. This is similar to this, that when we are getting tall and dwarf, here also we are getting tall and dwarf. That means the genotype of this plant is heterozygous dominant. Okay, genotype of this plant is heterozygous dominant. Then we call it, this cross is known as the test cross because we are crossing it with the homozygous recessive parent. Remember this. In case of the back cross, F1 hybrid is crossed with any of the parent. It could be crossed with the homozygous recessive parent, or it can be crossed with the homozygous dominant parent. Then this cross is known as a back cross. Okay, and please remember one point: in case of the test cross, the ratio will always be in monohybrid 1:1. You can see here is 50% tall and 50% dwarf. That means probability of this will be 1/2. Here also it is 1/2. So the ratio will be 1:1. Okay. Next one is a dihybrid cross. Di means 2. So in this case, we study 2 characters. So 2 characters will be studied, or the inheritance of the 2 gene. There are the 2 genes. There's Y gene and the R gene. Y stands for yellow and R for round. Here, small y represent the green color and small r represent the wrinkled. Okay. So in dihybrid cross, we study the inheritance of the 2 gene. So this parent will form a gamete capital Y, capital R, and this parent will form the gamete small y, small r. On the crossing, they produce a heterozygous offspring, which is yellow and round, but it is in heterozygous condition. That is capital Y, capital R, small r. So when this heterozygous will produce gamete, it will produce capital R, capital Y, capital R, small y, capital, small r, capital Y, and small r, small y. So the 4 gamete are produced. Remember, you can also calculate the gamete like this: 2 raised to the power n. So the number of gamete is equals to 2 raised to the power n, where n represent the number of heterozygotes. So in this case, how many heterozygotes are there? 1 and 2. So if 2 raised to power c equals to 4. So here also we are obtaining the 4 gamete. Okay. And one more important point: on the basis of monohybrid cross, the Mendel proposed 2 laws: law of dominance and law of segregation. Similarly, on the basis of dihybrid cross, Mendel proposed a law which is law of independent assortment. According to this law, when during the gamete formation, the alleles, they are assorted independently. Okay. For example, here you can see this is a gamete, this is a gamete number 2, this is 3, and this is 4. This capital R can go with capital Y or small y. They are independently assorted. This capital R is not influenced by capital Y, or capital Y is not influenced by capital R. So during the gamete formation, these alleles, they are assorted independently. And remember that law of independent assortment is also not a universal law, because exception of law of independent assortment is linkage. So when we now we do the selfing of F1 generation. So this is crossed with capital Y, small y, capital and small. On selfing, it produces 16 progeny, and they show a phenotype ratio of 9:3:3:1. And the genotypic ratio is 1:2:1. 2:4:2 or 1:2:1. Remember the phenotypic and the genotypic ratio of the dihybrid cross. Okay. Now moving next is the law of inheritance. First is the law of dominance. Second is the law of segregation and the law of independent assortment. Law of dominance state that in case of the heterozygous condition, the dominant allele will express and the recessive allele will not express. Okay. So this is a law of dominance. Law of segregation that during the gamete formation, one gamete will carry only one allele. So this is a law of segregation. Law of independent assortment says that during the gamete formation, the allele, they assorted independently. Next one is a non-Mendelian inheritance. These are the exceptions of the Mendelian inheritance. First one is the incomplete dominance. In incomplete dominance, the F1 progeny is in between. Or we also call it intermediate. So as you can see in the figure, there's a parent, the parent is red. One parent is having a red phenotype and another parent is having a white phenotype. So red is dominant and white is recessive. So it will produce a gamete I, it will produce a gamete W. So when they cross, they produce a pink color, which is an intermediate. That means the F1, generally in case of dominance, you have observed that if there's a heterozygous condition, in this, we are getting a top. That means if I cross with capital T, these are the parents. If I cross, they will produce gamete capital T, it will produce gamete small t. So their F1 will produce capital T and small t, right? This is what we are observing in case of dominance. This capital T and it is tall. That means the F1 progeny is resembling one of the parent. Here, this parent is tall. So the F1 and the progeny is also tall. So this F1 progeny is resembling one of the parent in case of dominance. But in case of incomplete dominance, you have observed that we are obtaining the intermediate. That is why it is called as incomplete dominance. And when this pink flower is self-pollinated, we are obtaining one red, two pink, and one white. Okay, so in case of incomplete dominance, the genotypic ratio is 1:2:1, and the phenotypic ratio is also 1:2:1. Okay, that means so remember, in case of incomplete dominance, you are observing the both that the phenotypic and the genotypic ratio, they are same. Okay, phenotypic is 1:1, 1:2:1, that is red, 2 pink, and 1 white. This is the phenotypic ratio, and genotypic ratio is also 1:2:1. So the phenotypic and the genotypic ratio are same in case of incomplete dominance. Now, first, let's discuss the multiple allism. Multiple allism show when there's a one gene, and this one gene is having a multiple alleles. For example, ABO blood group. Here, the gene I and this I genes is having a 3 alleles. There's IA, IB, and small I. Small I is recessive, while IA and IB are dominant. So, if, for example, if this person is having a blood group A, this person is having a blood group B. Let's keep a homozygous dominant condition. This person is having a genotype IA, this person is having a genotype IB. They will produce a gamete IA, this will produce a gamete IB. On crossing, it will produce IA and IB, that is the F1 generation. So in F1 generation, we are getting the AB blood group. IA and IB, because the genotype consists of IA and IB, so the blood group is AB because both IA and IB are dominant. So in case of codominance, remember this ABO blood group is also an example of codominance. So in case of codominance, the F1 progeny. This F1 progeny is resembling both parents. It's resembling both parents because it's showing the blood group AB. And the one parent is having a A blood group, one parent is having a B blood group. So this progeny is resembling both the parents. That is why it is an example of codominance. Okay. So please remember this, that in case of incomplete dominance, the F1 progeny is showing the intermediate. Okay. In case of dominance, the F1 progeny is resembling one of the parent. In case of codominance, the F1 progeny resemble both the parents. Okay. Now let's discuss the polygenic inheritance. In poly means many, right? Poly means many and genic means gene. So when many genes are controlling the one trait, then it is known as a polygenic inheritance. Skin color. Skin color is a type of polygenic inheritance. And a polygenic inheritance is also an example of quantitative inheritance. Why the quantitative inheritance? The more the dominant allele, the more is the expression. Okay. Next one is pleiotropy. Pleiotropy, when one gene controls many traits, then it is known as pleiotropy. Example, phenylketonuria. Okay, example of a pleiotropy. Or sickle cell anemia is also an example of pleiotropy. As you can see in this image, this ABO blood group, this is an example of multiple allism. That this ABO blood group, it shows multiple allism. Along with that, it also shows codominance. The skin color. Skin color is shows polygenic inheritance because there are many genes. There are A gene, B gene, and C gene, and they're controlling the skin color. So more the dominant allele, more is the expression. As you can see, this is very dark. Why? Because they're having a 6 dominant allele. This person is very light because there's no dominant allele. So more the dominant allele, more will be the expression. So it is quantitative inheritance. This is because the quantity of the dominant allele is determining the expression of the allele in the phenotype, expression of the gene in the progeny. For example, this is after crossing, they produce mulatto, which is an intermediate. They're having only 3 dominant allele. Okay. So it is less dark as compared to the parent. Now, this is an example of pleiotropy where one gene is controlling many traits. For example, this is a gene okay? If the defect occur in this gene, it leads to blindness, it leads to liver failure, it leads to heart attack. So that means the one gene is performing many function or controlling many function. If the defect occur in these genes, they lead to many disease, like it leads to blindness, it can lead to liver failure, it can lead to heart attack. So this is an example of pleiotropy. Next one is the chromosomal theory of inheritance. As we know that the Mendel work was not recognized earlier, but De Vries, Correns, and von Teschemack, they independently rediscovered the Mendel's result. Okay. And chromosomal theory of inheritance was proposed by Sutton and Boveri. Please remember this, that the chromosomal theory of inheritance was proposed by Sutton and Boveri, and the experimental verification for the chromosomal theory of inheritance was proposed by Morgan. Okay, it's very important, remember this. So Sutton and Boveri, they proposed that the chromosome and the gene, they show a close similarity. Like during the gamete formation, as the chromosomes get separated, similarly, the genes which are present on the chromosome, they also get segregated. See, this is a G1 phase. Okay, in this, the cell in the G1 phase, the cell undergo enlargement, increase in size, right? After G1 is the S phase, where the DNA duplication or the centrosome duplication takes place. S phase is followed by the G2 phase. You can see now the DNA duplication has occurred. After G2 phase is the M phase. Here's the meiosis. So first, let's discuss the meiosis 1 anaphase, where the homologous chromosomes, they get separated. Homologous chromosomes, the pair of the homologous chromosomes, they get separated, and the two identical chromosomes, they form the homologous pair. So in meiosis 1 anaphase, the homologous pairs, they get separated. In meiosis 2 anaphase, the sister chromatid gets separated. So we are getting the 4 haploid daughter cells. So as the chromosomes get separated during the gamete formation, the genes which are present on the chromosome, they also get segregated. So this is the chromosomal theory of inheritance. And the experimental proof for the chromosomal theory of inheritance was proposed by T.H. Morgan. Remember this. T.H. Morgan carried out the experiment on Drosophila and provide the experimental verification for the chromosomal theory of inheritance. So he worked on Drosophila, and why he chose Drosophila? Because it is easy to grow. The characters are easily distinguishable in the Drosophila, and they have a small chromosome size, and they produce a large number of offspring in one mating. And the males, they are distinguishable from the female. Like male are smaller in size and the female are larger in size. Okay, and they have a very short lifespan of the short generation time. That is why T.H. Morgan worked on Drosophila. T.H. Morgan also worked on linkage. Sorry, T.H. Morgan proposed a term that is linkage. Linkage shows that there's a physical association between the gene. And the linkage, they are of two types. There's a complete linkage or incomplete linkage. In case of complete linkage, we observe 100% parental and no recombination. That means no recombinant means no non-parental type. Recombinants are also known as non-parental type. So if there's a complete linkage, okay, that means even after the crossing over, we are getting the 100% parental progeny and no recombinants are obtained. That is the case of a complete linkage. That means the genes are completely linked and they cannot be separated during the crossing over. Incomplete linkage, when we are obtaining the parental as well as recombinants. If we are getting the recombinants in the offspring, that means it's an incomplete linkage. Remember this, that the percentage of parental would be more than 50 and the percentage of recombinant would be less than 50. That is, the recombination frequency cannot exceed 50. Please remember this. And the incomplete linkage is further of two types. There's a tightly linked or loosely linked. As you can see in this image, this is a tightly linked and the loosely linked. So, for example, if this is an incomplete linkage case, okay, we are getting in one cross, we are getting 75% parental and 25% recombinant. Okay, I'm giving a hypothetical situation. And in the other cross, you are obtaining parental 95% and recombinants 5%. Because we are obtaining the recombinant, that means it is the case of incomplete linkage. But here, we are obtaining the more recombinant. The percentage of recombinants are more, that is 25%. Here, the percentage of recombinants are 5%. So this could be a case of loosely linked. That is, the distance between the two genes are more. And this is the case of tightly linked. That is, the distance between the two genes are less. So linkage is inversely proportional to recombination frequency. And linkage is also inversely proportional to the distance between the genes. So more the recombination frequency, less will be the linkage. They are inversely proportional. Okay. And Mendel and Morgan carried out 3 dihybrid cross on Drosophila, and he chose 3 characters: there's body color, eye color, and the wings, whether it's normal wings or the miniature wings. So he performed the 3 cross. See, in the first cross was done between the body color and the eye color. And he is obtaining parental 98.7% and recombinant 1.3%. That means we are obtaining the recombinant in this case. That means this is an example of incomplete linkage. Second cross he done between the wings and the eye color. And he obtained the parental 62.8% and the recombinant 37.2%. Okay, so here also this is an example of incomplete linkage. So first cross is done between the body color and the eye color. We are getting the recombinant is 1.3%, right? Second cross is done between wings and the eye color. We are getting the recombinant 37.2%. And third cross is done between the body color and the wing size, and we get the recombinants okay. So there was a student of Morgan, which is the name of the student is Alphonse Toutwent. So Alphonse Toutwent, he proposed gene mapping. He said that the frequency, the recombination frequency can be used as a distance between the genes. So the recombination frequency is used as a measure of distance between the genes. And through this, he proposed gene mapping. His name was Alphonse Toutwent. So remember, if the question asks you about the linkage, then linkage was proposed by T.H. Morgan. If they ask you about the gene mapping, it was proposed by Alphonse Toutwent. So he said that we can also write this recombination frequency as 1.3 centimorgan. That is the distance between the gene. That is, so here the 1.3 centimorgan represent the distance between the Y gene and the W gene. That is the gene of the body color and the gene of the eye color. Okay. Now we can map the genes on the chromosome. First, we'll go with the the cross which is having a highest recombination frequency, that is 38.5%. So the distance between the body color and the wing is 38.5%. Centimorgan. Now we'll go with the least one. So the distance between the eye color and the body color is 1.3 centimorgan. Now you can easily calculate the distance between the eye color and the wing is 37.2 centimorgan. Okay, so first is body color, then followed by the eye color, then followed by the wings. So this is how we can map the gene on the chromosome. So gene mapping was proposed by Alphonse Toutwent. Next one is the mechanism of sex determination. This was the foundation of this mechanism was led by Hanking, and Hanking, he worked on insect, and he observed that during the spermatogenesis, he observed the nuclear structure, that 50% of the gamete is carrying the nuclear structure. And he called these nuclear structure as X body, but later the other scientists, they find out that this X body is the X chromosome. So first, these two mechanisms, they are showing that the male are heterogametic. Hetero means different, that they produce different gametes. This is mechanism found in humans and Drosophila. This XXXO is found in case of insects. Grasshopper, example grasshopper, where the female is homogametic and male are heterogametic. That means the one gamete will carry the X chromosome and the one gamete will carry the Y chromosome in case of humans and Drosophila. So the ZZ and ZW is where the female is heterogametic. Here the female is heterogametic, so that means the males are homogametic. And this type of mechanism, ZZ and ZW, seen in case of birds. So in this case, the 50% of the gamete will carry the Z chromosome, 50% will carry the W chromosome. So the female are heterogametic. Next one is the sex determination in case of bee, and they follow a haplodiploidy condition, or the haplodiploidy mechanism. Here the ploidy will determine the sex of the offspring, whether it's going to be queen or the drone. The queen is having a 32. This will undergo meiosis and will produce so if the egg without fertilization through parthenogenesis is converted into the offspring, that is in this case, in case of bee, they are the male and they are drone. They are known as drone. If this egg is fertilized, develop into the zygote will develop in the offspring. If this offspring is feed on royal jelly, it will lead to a queen. And here the 2n equals to 32. Remember, because the zygote is 2n equals to 32. And if this offspring feed on bee bread, then it will form worker or the sterile female. Okay, so here the ploidy is deciding whether it's a male or the female. Here this is haploid, haploids are male and diploids are female. Now mutation. Mutation are the sudden changes in the gene sequence or in the DNA sequence. For if there is a single base pair change, for example, here's T and T is replaced by C. So there's a single base pair change, then it is known as a point mutation. An example of point mutation is a sickle cell anemia. But if there's a deletion or insertion, then it will lead to a frame shift mutation. Frame shift mutation is the change in the reading sequence. Example, this is the original sequence. Okay, if there is a deletion occurring in this T is deleted. Now the entire reading frame has changed. Earlier it was ATG and GTC. Now this T is removed. Now it has become AGT and GC. Okay, so this type of mutation is known as a frame shift mutation. Next one is the classification of the mutagen. There are different types of mutagen. It could be a physical mutagen, chemical mutagen, or biological mutagen. Physical mutagen, there are gamma rays, X-rays, UV rays. Chemicals, they are the alkylating agent, deaminating agent, intercalating agent. Biological, they are biological agents, could be like virus, bacteria, or transposomes. Next one is the pedigree analysis. Please remember this. The symbols and what these symbols represent, please remember this because it's important. They might give you the symbol and they can ask you what does it represent by giving you the option. In pedigree analysis, you will study there's autosomal dominant, there's autosomal recessive, X-linked recessive, X-linked dominant, and Y-linked. In case of Y-linked, you have observed that only male will get affected because only the males they have Y chromosome. So in Y-linked, you will see that only the male will get affected. And the major difference between the dominant and the recessive is the skip of generation. In case of recessive, you can see the skip of generation. Here, no one is affected. But here you can see that this offspring is, this female is affected. So there's a skip of generation. But in case of dominant, you can see there's no skip of generation. Here the parents are also affected and the offsprings are also affected. Okay. Next one is the genetic disorder. First, we are going to discuss the Mendelian disorder like hemophilia. Hemophilia and the color blindness, they are X-linked recessive disorder. Remember, that hemophilia and color blindness are X-linked recessive disorder. Thalassemia is an autosomal recessive disorder. Sickle cell anemia is an autosomal recessive disorder. Phenylketonuria is also an autosomal recessive disorder. Cystic fibrosis is also autosomal recessive disorder. But myotonic dystrophy is an autosomal dominant disorder. Remember this. In case of hemophilia, the person which is affected by the hemophilia, they lack the proteins which are involved in blood clotting. In case of color blindness, the person which is suffering from color blindness cannot able to distinguish between the red and the green color. See, sickle cell anemia is also an example of point mutation and it is an autosomal recessive disorder. See, in mRNA, there's GAG, and in this case, there's GUG. Because of the point mutation, because this T is replaced by A, so we are getting in the mRNA, because mRNA is the complementary of the template strand. So in mRNA, we are getting GUG. And this codon will code for valine at the 6th position. In the original sequence, this GAG codes for glutamic acid at the 6th position. But after the mutation, the codon form is GUG, and this codon codes for amino acid, which is valine. Okay, so this is an example of sickle cell anemia because the shape of the RBC gets changed. It becomes sickle shaped. Phenylketonuria is also an example of pleiotrophy. Here, what happened? The person suffering from phenylketonuria, they lack the enzyme which is phenylalanine hydroxylase, because this enzyme is convert the phenylalanine to tyrosine. If this enzyme is absent, this phenylalanine cannot be converted to tyrosine and it will convert it into phenylpyruvic acid, which get accumulated in the body and can lead to mental retardation or the formation of ketone bodies. Next one is thalassemia. Thalassemia is also an autosomal recessive disorder, and there are two types of thalassemia, alpha thalassemia and beta thalassemia. So in case of alpha thalassemia, there's two genes are involved. There's HBA1, HBA2, and both the genes, they are present on a chromosome number 16. Okay, so if the defect occur in these genes, the alpha chains will not be produced. Okay, if the production of the alpha chain will not occur, because we know that the hemoglobin, they have 2 alpha chain and 2 beta chain, because it's a quaternary structure. It has 4 subunits, 2 alpha chain and 2 beta chain. So if in alpha thalassemia, what happens if the mutation occur in these two genes, the alpha chains, they are not produced. So there's the beta chain, so there's a more production of the beta chain as compared to the alpha chain. And these beta chain, they form an unstable tetramer, because of which they produce the abnormal O2 dissociation curve. In case of beta thalassemia, the gene HBB, which is present on chromosome number 11, get affected. If this gene get affected, then beta chain will not produce. So automatically the production of alpha chain would be more, right? So these alpha chain they get accumulated on the surface of RBC, and they lead to the degradation of the RBC. So thalassemia, there are two types, alpha thalassemia and beta thalassemia. Next one is aneuploidy. So chromosomal disorder, they generally occurs to the aneuploidy. Aneuploidy is the loss or gain of the chromosome. Okay. And this aneuploidy occurs due to the non-disjunction of the chromosome. Non-disjunction means that the chromosome, they are failed to segregate. If they fail to segregate, that means the one gamete will have a gain of chromosome, or the one gamete will have a loss of chromosome. And euploidy, when there's a gain or loss of the complete set of chromosome. Next one is the chromosomal disorder. Number one is the Down syndrome. Down syndrome occurs because of the trisomy at 21 chromosome. Trisomy of 21 chromosome leads to a condition which is known as Down syndrome. Turner syndrome, when there is a loss of X chromosome. Klinefelter syndrome, when there is a extra copy of X chromosome is present in case of male. This is observed in case of male. This is Turner syndrome is observed in case of female. And the symptoms of Down syndrome, they have a furrowed tongue, they have short-heighted, they have mental retardation. Okay. And remember one more, there's an Edwards syndrome and a Patau syndrome. Edwards syndrome, when the trisomy occurs at 18 chromosome. And Patau syndrome, when the trisomy occur at 13th chromosome. So that means in Edwards syndrome, there's an extra copy of chromosome number 18, and in Patau syndrome, there's an extra copy of chromosome number 13. Remember this. Moving next is the question. Question number 1 says, given below are the two statements. Statement 1 is Mendel studied 7 pair of contrasting traits in pea plant and proposed the laws of inheritance. Statement 2, that the 7 character examined by Mendel in his experiment on pea plants were seed shape and seed color, flower color, pod shape, pod color, flower position, and stem height. In the light of the above statement, choose the correct answer from the option given below. What do you think? What will be the correct option? They have given you the two statements, right? Please remember the 7 characters. Please remember all the traits. Please remember that which trait is dominant and which one is recessive. Okay, these are very important. And please remember the chromosomal syndrome. Please remember the whether it's a Mendelian disorder, and also please remember whether it's an autosomal dominant, whether it's an autosomal recessive or an X-linked recessive or X-linked dominant. Please remember these. These are very important. Quickly answer. What do you think? What will be the correct option for this question? So statement 1 says the Mendel studies 7 pair of contrasting traits. Yes. 7 pairs means 14 traits. Yes, Mendel studied 14 traits. And proposed the laws of inheritance. Yes, he proposed the law of inheritance. 7 character examined by Mendel on the pea plants were pea seed shape. Yes, color, flower color, pod shape, pod color, flower position, and stem height. Yes, 7 characters were studied and 14 traits. So both statements are correct. So both statement 1 and statement 2 are correct. So option would be 4th. Moving next is question number 2. The number of contrasting character studied by Mendel for his experiment was. They're asking about the number of contrasting characters. So the number of contrasting characters studied by the Mendel for his experiment is 7. Next one is the experimental verification of the chromosomal theory of inheritance was done by. They're asking about the experimental verification, I told you. Was done by. So chromosomal theory of inheritance was proposed by Sutton and Bovie. But the experimental verification is done by Morgan. Remember this. So the correct option is 1, Morgan. Mendel worked on pea plant. Sutton and Bovie, they proposed the chromosomal theory of inheritance, and the experimental verification of the chromosomal theory of inheritance was done by Morgan, and he worked on Drosophila. Next question says in Snapdragon, a red flower was crossed with a white flower, and if F1 generation, the pink flower were obtained. When the pink flower was saved, the F2 generation show white, red, and pink flower. Choose the incorrect statement from the following. First, there's a red flower crossed with white. This will produce a gamete. So in F1 progeny, we are getting pink, which is an intermediate. If we self this will produce 2 gametes. This will also produce 2 gametes. That means we are getting red, we are getting pink, and we are getting white in F2 generation. First option says that this experiment does not follow the principle of dominance. Yes, because this is a case of incomplete dominance, right? Pink flower in F1 is due to incomplete dominance. Yes, pink flower in the F1 is due to incomplete dominance. Ratio of F2 is 1/4 red. Yes, it is 1/4 red. These are 2/4. And this is white is also 1/4. So this is also correct. 4 says the law of segregation does not apply in this case. This is wrong. Law of segregation is a universal law I told you, and law of segregation is applicable here because the one gamete will carry only the one allele, right? So the law of segregation is applicable here, but the fourth statement option says that the law of segregation does not apply in this experiment. And the question is asking about the choose the incorrect statement from the following. So it is fourth. So incorrect statement is fourth. I hope it is clear to everyone. So it's already 1. We are winding up the class. Thank you all for your participation. And see you in the next class.